Good day to our lecturer. This is a group presentation by Group H, the microbial discovery to market failure case of plazomicin. This is the task distribution for all of the group members.
First, I will introduce the biological lead of plazomicin. The biological lead of plazomicin is cecomycin, which is a naturally occurring aminoglycoside antibiotic. It's derived from Macromonospora ionescens and marine streptomyces species. As a member of the aminoglycoside class, cecomycin exhibits strong antibacterial activity, especially against gram-negative bacteria. In terms of its mechanism of action, cecomycin binds to the A site of the 16S ribosomal RNA, thereby inhibiting protein synthesis. It can also cross the outer membrane of gram-negative bacteria by disrupting mechanism ion bridges. The combined action of these two mechanisms constitutes the bactericidal effect of cecomycin. However, despite its effectiveness, cecomycin has a major limitation. It's highly susceptible to inactivation by aminoglycoside modifying enzymes, or AMEs, such as AAC, ANT, and APH, which contributes to antibiotic resistance. To overcome this problem, chemical modifications were introduced to transform cecomycin into plazomicin. As shown in the figure, the addition of a HABA substituent at position 1 and a hydroxyiso group at position 6' helps protect the molecule from enzymatic inactivation. As a result, the final product, plazomicin, retains the bactericidal activity of aminoglycosides while showing enhanced efficacy against the multi-drug resistant gram-negative pathogens. Next, Tang will introduce the mechanisms of action of plazomicin.
Good day everyone, my name is Tang Yusim and I'll be talking about the mechanism of action of plazomicin. Plazomicin is a positively charged aminoglycoside antibiotic. It will enter electrostatically through the negatively charged cell membrane of the bacteria cell. And then it will enter through the inner membrane via protein pumps. As it enters the cytoplasm, the plazomicin will bind specifically to the A site of the 16S rRNA within the 30S ribosomal subunit. It will then interfere with the binding of tRNA carrying the amino acid to the codon of mRNA. Additionally, it will interfere with the initial amino acid formation, leading to mistranslation. Finally, it increases the permeability of the cell membrane, which will then lead to cell death.
Thank you, Tang. Now I'll talk about the discovery of plazomicin. Firstly, the name plazomicin was given by the producing company, Achaogen, and initially it was known as ACHN 490. As shown earlier by Ho, the chemical structure of plazomicin consists of hydroxyaminobutyric acid and cecomycin. The final molecule has a chemical formula of C25H48N6O10, a molecular weight of 592.68, and consists of 490 cecomycin analogs. This gives rise to the name ACHN 490. Moving on to the timeline of events, not much of the discovery itself was disclosed, but what was known is the development of plazomicin was initiated in 2009 by Achaogen. In the following years, the drug entered clinical trials, Phase 2 from 2010 to 2012, Phase 3 from 2013 to 2016. In 2018, the drug was finally approved by the FDA for the treatment of complicated urinary tract infections, CUTI, and after the bankruptcy of Achaogen in 2019, the rights to plazomicin were acquired by Cipla and sold under the trade name Zambri. Today, Zambri is sold as an injection-based intravenous aminoglycoside antibiotic. It is a non-controlled drug available as prescription only and is generally advised against using unless necessary. Now I'll pass to John to elaborate on the hype of plazomicin.
Thank you, Zheng Wei. I'm Yu Hao. In part 4, I will explain the massive hype around plazomicin. First, an urgent medical need. Plazomicin targeted the CRE, widely known as nightmare bacteria. By successfully evading bacterial enzymes, it offered a true scientific breakthrough against these deadly superbugs. Second, unprecedented regulatory backing, Plazomicin made history as the first-ever antibiotic to get the FDA's breakthrough therapy designation. It also secured millions of dollars in funding from US BARDA. Finally, huge market confidence. Its first 3 trials showed great results compared to older drugs. This sparked strong investor interest and led to a highly successful IPO for its developer, Eccogene. Despite this massive hype, it crashed. I will now pass to Tiantian to explain why.
Now we move to the part 5 of our case, the failure of plazomicin. So why did plazomicin fail? The first blow was regulatory. The FDA rejected its use for several bloodstream infections, approving it only for complicated UTIs. This was a major setback, as the UTI market is already flooded with cheap generics, leaving plazomicin with a very narrow, last resort patient pool. Next is the antibiotic paradox. The numbers simply didn't work. Occasion spent $1 billion on development, but first-year sales were only $1 million. Hospitals refused to pay $1,000 for a new drug when $20 generics were good enough. Simply put, the cost of innovation far outweighed the market's willingness to pay. Thirdly, Occasion faced the clinical inertia. Doctors are often cautious with this class of drugs due to historical toxicity concerns. As a small biotech, Occasion also lacked the massive sales force of Big Pharma to change these prescribing habits or compete with established brands. The result was catastrophic. Just 10 months after FDA approval, Occasion filed for bankruptcy. The rights to this breakthrough drug was sold for only $16 million, a tiny fraction of its cost. Plazomicin's story proves that even perfect science cannot survive a broken antibiotic market.
Moving on to the part 6.1, reflections on the failure, which is the case of plazomicin, demonstrate both scientific and also healthcare system challenges in sustaining new antibiotics. The first point is antibiotic stewardship limits market demand. Antimicrobial resistance or AMR occurs when microorganisms become resistant to antibiotics, making infections harder to treat. To address this, antimicrobial stewardship programs were introduced to promote responsible antibiotic use and reduce the spread of resistant bacteria. However, this also means that new antibiotics like plazomicin are only used in severe cases. As a result, prescription rates are very low, which reduce market demand and limit revenue for the drug. Secondly, high cost and risk of antibiotic development. Antibiotic development is both costly and high risk. Many pharmaceutical companies have withdrawn from this field due to scientific, regulatory, and financial challenges. In addition, antibiotics are typically used for short treatment duration, resulting in lower returns compared to other drugs. Developing antibiotic also requires very long research timelines and expensive clinical trials. This creates significant financial risk, especially for smaller companies like Ekeogen. Next is scientific success versus market sustainability. The case of plazomicin shows that scientific success does not guarantee market sustainability. As larger pharmaceutical companies exited antibiotic research, smaller biotech companies took over with limited resources. Although the drug was effective against resistant bacteria, it faced a very low demand and commercial challenges. This led to poor sales and eventually the bankruptcy of Akeogen in 2019.
Thank you, Nazira. Now, after understanding the scientific and healthcare challenges, let's move to the bigger picture reflections on the economy and policy strategies needed to sustain antibiotic innovation. First, we need deadline payment models. Like the UK's NHS subscription pilot, this separates revenue from sales volume, ensuring that drugs reserved for a rare emergency remain financially viable. Second, public funding must come with strings attached. Presomyzin was largely developed using US taxpayers' dollars. Yet, after bankruptcy, its rivals sold overseas without patient access guarantees. Future public investment must require that if taxpayers fund the research, the drug must be available to those who need it. Third, we must restore investors' confidence. Alcatra's bankruptcy sent a chilling message that novel antibiotics have zero commercial value. To break this cycle, we need successful asset stories, acquisitions, or IPOs to prove antibiotics can be a viable business again and encourage big pharma to re-enter the field. These strategies are not just theoretical, they are urgent calls to action. To conclude, Presomycin's story is a wake-up call. Without sustainable economy models, scientific breakthroughs alone cannot save patients. Thank you for listening. This is Group H presentation.
These are all the references. Thank you.